Well, good morning, everyone, and welcome to Emerald Ash Borer University. This is a webinar series that's funded by the USDA Forest Service, and we thank them for their help. My name is Robin Osborne, and I'm coming to you from Michigan State University. Along with my EAB University colleagues, Amy Stone from The Ohio State University and Dr. Cliff Sadoff from Purdue University, we welcome you to today's presentation by Dr. Jennifer Koch, research biologist for the USDA Forest Service in Delaware, Ohio. She will be presenting information regarding Beech Bark Disease and the efforts to find and cultivate host plant resistance to it. Jennifer has both a master's degree and a PhD in molecular genetics from The Ohio State University. In her position at the USDA Forest Service, she is a working member of the Restoration and Conservation of Rural and Urban Forests Research Unit. She is interested in using genetics and breeding to manage invasive insects and pathogen threats through the development of host tree resistance. Her research is currently focused on developing resistance to beech bark disease in the American beech and resistance to the emerald ash borer and North American species of ash. Before we get started, I want to remind you that your comments and questions are welcome today. Please feel free to write them in the question and answer feature, which you can find by mousing over the bottom or occasionally it's the top of your screen. We will be making a note of all the questions, and Jennifer will respond to them after the presentation so we can keep the webinar flowing smoothly. To keep these free webinars coming, we need your feedback. There will be a link provided to you in the Q&A pod at the end of the webinar for a short voluntary confidential survey that I hope you will take time to fill out. If you're one of the first 10 people to fill it out, we'll be sending you an emerald ash borer goodie bag. But either way, we hope that you will give us feedback. I will also be sending out an email after the webinar to you with that link in case you weren't able to access it during the webinar. For those of you who want CEUs, if you would like a certificate indicating that you participated in today's live webinar, complete the survey and send an email message to Amy Stone at stone.91@osu.edu. Certificates will be mailed to you within a week of today's program. The webinar is being recorded today and will be available for viewing soon at www.emeraldashborer.info. You will also find the recordings for all our previous EAB University webinars there. And with that, thank you for participating today. And Jennifer, I see you've got your screen up, so you can start your presentation.
Thanks, Robin. Thanks for everyone for tuning in today. I'm looking forward to sharing our work on utilizing host resistance to combat beech bark disease. I wanted to start off by talking about the importance of American beech. When I started working on this species over 10 years ago now, sometimes I'd interact with foresters who would ask, why do we care about beech bark disease? Let's just get rid of beech. It's a nuisance. But the reality is it is quite an important species. It provides food and habitat for over 40 different species of birds and mammals. And there's been studies that have shown that when beech nut production is down, it impacts black bear birth rates as well as the rate of survival of their cubs. And in some northern hardwood forests, beech is now the only mass-producing species, so it's particularly important there. So forest managers are interested in managing, those that are interested in managing for wildlife typically have a great interest in managing to maintain healthy American beech as part of their forest. So moving into beech bark disease, just to give a little background in case there's anyone who's not very familiar with it, this disease is a causal complex. So it's caused by two different entities, the first being an invasive beech scale insect that's shown on the left-hand panel. It's a soft-bodied scale insect that, if you look down in the lower left, only has one mobile phase in its life cycle that occurs around here in between July and August. So it is primarily dispersed through wind and with people and hitching a ride on various animals. And on the right-hand side of the panel, you see the Neonectria fungi. This is the fruiting bodies. The feeding by the beech scale insect creates tiny little cracks in the bark that allow points of entry for this fungal infection to occur. There are 2 species of Neonectria fungus that are associated with beech bark disease. Neonectria datissima is actually a native species, and Neonectria faginata, although it's only found in North America, it's more closely related to exotic species than to any of our native species. So the current theory is that it potentially was derived from an introduced species. But the point being that these fungi do not or have not been associated with massive disease and mortality without the presence of the scale insect. So this slide shows the insect and the fungus at a more macroscopic level. On the left-hand panel, you can see the typically whitewashed appearance that the scale causes trees to have, and that is a result of this white wax-like substance that they secrete. And on the right-hand side is a larger view of the Neonectria infection. Neonectria usually causes small cankers, but when they are enough of them, they will begin to coalesce, and that results in girdling the tree and eventually can lead to mortality. There have been 3 stages of beech bark disease defined that are typically referred to. The first is the advancing front, and that is to describe a situation where beech scale is present in the population, but there's not any Neonectria infection that has yet occurred. There's also a killing front, and this is describing once the scale populations have increased and they're very abundant, and Neonectria infection is now present, and you're starting to see high levels of mortality. Mortality begins about 3 to 6 years after Neonectria, well, and many times after the scale population is there, but definitely once Neonectria infection occurs, and this can vary. The aftermath forest describes the situation once the more mature, most susceptible trees have died, and the beech scale population is now, it's still there at a residual lower level. And you see a shift from the larger diameter beech into the smaller diameter beech that occurs with the loss of the most mature trees. Remaining trees that are present are shown in the picture on the right-hand side. They're typically deformed. A lot of the cankers become very deformed as the tree tries to defend itself from the fungal infection. And there are a few large trees that will persist that are potentially resistant. This slide is showing the direction of the beech scale invasion. Since its introduction in about 1929 in Maine, it actually came in on a European beech and has slowly spread through New England ever since. It is estimated to spread about 6 miles per year, although you can see the outbreak in 1993 down in the Smoky Mountains, and then in 2000 up in the lower peninsula of Michigan were both sort of outlier infestations. And so those outliers, instead of being part of the advancing front, are believed to have been caused by human movement, mostly because both of those are very highly popular tourist attractions. Ludington State Park in Michigan is the highest traveled state park in Michigan, and that's where we first identified scale in that state. The green shows the range of American beech in the United States. So the good news is that beech bark disease has not made its way through, although it is anticipated that it will continue to move throughout the range of beech. The red shows where full-blown beech bark disease is present, meaning the scale and the fungus has been identified. And then if you look at the blue areas, that is depicting that advancing scale front. Just to give you an idea of what the impacts of this disease are at the different stages, these photos were taken of the killing front in Ludington State Park. And the picture on the left shows a beech tree that you can see the white is covering most of the bark, the scale insect. But you can also clearly see a dark strip of going down the length of the bole. And that's where the bark has been killed by Neonectria infection. So it's once the bark is dead, it's no longer suitable habitat for the scale insect. So that's why there's no scale on this particular part of the tree. But that long strip of dead bark obviously is making that tree vulnerable. So if you look at the middle photo, it's not uncommon to see what we refer to as beech snap. And this is when wind stresses the tree even further and causes it to break. So beech bark disease is causing a lot of fallen trees, and hazard trees are created. The photo on the far right is showing a heavily cankered survivor tree. You can see that this tree is damaged, it's stunted in growth, so it has no economic value. It also has very little value for wildlife. This photo is showing, again, Ludington State Park at the killing front. In the first wave of the disease, it's estimated that mortality can reach somewhere between 50 and 80% in most stands. You can see in this picture a canopy opening that's been created by the death of several large beech trees in this area. In the aftermath forest, one of the things that is common to be seen is this formation of what is called a beech thicket. And this happens when the diseased trees begin to throw off root sprouts quite extensively in a response to the disease, sort of a last-ditch effort to survive. In this photo, all of the brown leaves are from a single source mother tree that has been cut, and that stump has been treated with insecticide. So all of the connected root sprouts, not insecticide, herbicide, all of the connected root sprouts are turning brown from that herbicide treatment. So you can see it's pretty extensive just from a single tree. And this type of a thicket is going to prevent regeneration of other species. And again, it is not going to have the wildlife that value that a healthy, mature American beech tree would have with the production of mast and habitat. I had mentioned that sometimes we do see symptom-free American beech trees that exist in areas where there has been a long history of beech bark disease infestation. And in 1982, Dave Houston actually published a paper where through artificial inoculation, and a picture of artificial inoculation with the insect is shown in the bottom right, demonstrated that the resistance is to the scale insect and not to the fungus. So when you inoculate with fungal bark plugs, you can cause a fungal infection, but without the scale causing all of the entry wounds into the bark, typically the fungus is not an issue. It's estimated that somewhere between 1 and 5% of beech trees are resistant, and this is of course going to vary from population to population. The bottom left-hand corner shows a cluster of resistant trees, and it's not uncommon for these trees to be found in clusters, which was an important first clue that the resistance is likely a genetic trait and not a result of a particular environment, because trees that are growing close to each other, especially beech trees, are likely either clonally related root sprouts or full or half seedlings. So if you think about it, many people might be wondering, how is it possible that a native tree species has resistance to a non-native invasive insect, and this is possible even with pathogens, that it has never been exposed to before and hasn't had the time or the opportunity to develop resistance through coevolution? Well, the answer to that lies in the tremendous amount of genetic diversity that can be found in natural stands of hardwood trees. They're open pollinated, lots of diversity. So that means that there's a lot of different alleles and combinations of alleles found throughout any population. So there can be rare alleles that can confer resistance or tolerance even when there is not coevolution. And these are going to be rare because there's no selective advantage that would make them represented more frequently in the population because the invasive has not yet arrived. So the key is actually to increase population level frequency of resistance to manage the invasive species threat. And in this next series of slides, I'm going to try to depict the concept of enriching the population for genetic resistance. And so in this example, in this cartoon stand, natural stand that's depicted here, I've shown about 5 out of 26 of these trees, or 17%, are shown that are resistant. So there's a lot of diversity in this stand, but a low level of resistance. At 17%, that's higher than what we'd expect with Beech Bark Disease, and way higher than what we'd expect with a lot of other pathogens that have been studied. So your invasive insect or pathogen comes in and attacks the susceptible trees, and it's going to kill some of those. Some of them are going to be missed just as escapes, and maybe later on they'll be infested. But the result is, as those trees die off, this population has now been reduced to a smaller number of trees. The same 5 resistant trees are still there, but now we only have 5 susceptible trees left. So 5 out of 10 trees, we've gone from 17% resistance in the population to a proportion of 50% in the population. So the population is now enriched for resistance. So now that there are a higher proportion of resistant trees in the population than there were before, the resistant trees will have a better opportunity to breed between each other, increasing the chance of the progeny they're producing being resistant. So now I've shown the next generation with an even greater number of resistant progenies. But if I back up to the natural stand, this selection of resistant trees, the way I've described it, is actually natural selection, when the pest or the pathogen is actually killing the susceptible tree. And in some cases, this can be effective if it's not killing so many of the trees that there are not enough resistant trees left in close enough proximity that they can breed with each other. And in some cases, the susceptible trees, like with Beech Bark Disease, a lot of those trees are lingering on. They're continuing to produce pollen, so they're continuing to contribute to the next generation. So they're continuing to create susceptible individuals. So we can kind of push natural selection along by doing one of 2 things. We can go in and physically remove, through silviculture, those diseased trees, and that will also lead to that enrichment in our breeding population. Or we can actually physically identify the resistant trees in the natural stand, and through grafting or seed collections, move them to a different location into a breeding program. But the outcome is still the same, that we have this breeding population enriched for resistance that can then, in the next generation, produce a population that's even enriched for resistance to a greater level. So in this example, I'm showing now 18 out of 26 trees. So in this generation, we're up to 70% resistance. And of course, if it's a breeding program, you're going to continue to test, and the susceptibles that have made it through will, through further generations, eventually be removed. Or in a natural population where you have continual pressure from the insect or pathogen, eventually those more and more of those susceptible trees will continue to die off. So the process that I was just describing, where you're selecting trees in a natural stand for a particular trait of interest and then going through a recurrent process of breeding and testing, is actually what's known as tree improvement. Tree improvement programs have been around for decades. And typically, especially when they were first started, the focus was on improving for traits involved in timber production. So the interest was in selecting for growth or form. But with this onslaught of invasives, I think that provides an argument for us to reinvigorate tree improvement programs and the infrastructure that it takes, so that we can apply those same principles and concepts to selecting and breeding for resistant trees, and not just focusing on timber production. So this slide is depicting that process, where in the first generation of selection, breeding, and testing, a seed orchard is developed, and that produces improved seedlings, much like how I tried to diagram in my cartoons just before. But you can also then select from those seedlings recurrently and then start a second generation. So again, in each generation, you're continuing to enrich for the trait of interest, being resistance in this case. So applied tree improvement or breeding is a great way to combat invasive threats by enriching host resistance. And again, this is not something that we're inventing. This is an old process. It's been around for a long time, but with new threats, we can apply those same methods. The genetics theories and concepts that have been being utilized through tree improvement programs have been proven to be successful. So this is an approach we know can work. And as a matter of fact, there's 255 programs worldwide that are currently involved in breeding for insect or disease resistance in forest tree species. So host resistance is actually providing a solution to invasives that is what could be termed a green solution, because we're not using insecticides, we're not talking about GMOs or transgenics, just accelerating nature through breeding select trees. The other good thing about breeding programs is that it allows us to retain genetic diversity. So when invasives are killing such a huge proportion of the population that there's very few members of the population left, this is going to decrease your genetic diversity. And that can lead to things such as inbreeding depression or genetic bottlenecks. But in a breeding program, you can take care to retain that genetic diversity and also retain the ability to adapt to the next threat, the next invasive insect or pathogen, or even the ability to adapt as climate change is impacting the environment. And also this genetic diversity is important for durable population level resistance and to just a generally sustainable, resilient population. So with our approach to beech bark disease, we are taking a traditional tree improvement approach using a breeding strategy. And so it's the same steps that were outlined in the previous slides of selecting from a diverse stand for resistance to beech bark disease, breeding, carrying out genetic studies to understand that it's how it's inherited, and design the breeding program that's needed to enrich the level of resistance, and consistent testing to constantly verify and select for the best parents, and all of this leading to establishing seed orchards, which can then produce enriched seed for outplantings. So the first step is selecting resistant germplasm. So just to touch on our program and how we're accomplishing that, we're very fortunate that we're working with a really strong network of collaborators, and our collaborators are responsible for the most part for this step, which is surveying for resistance. So the map that's shown here is from the Hiawatha National Forest. The areas in purple are areas where they have heavy beech stands. So when looking for resistance, we're going to focus on areas with a high amount of beech that we also know has been heavily impacted by beech bark disease. And our cooperators will then survey these areas looking for trees that have healthy crowns. We focus on trees with a minimum diameter at breast height greater than 9 inches. And of course, we want to identify any that are lacking any insect or disease symptoms. And these would be our putative resistant trees. Once these surveys are conducted, we train our partners on how to perform field screening to confirm that it is truly a scale-resistant tree. I've put a link up here for a video that we actually made, as well as a publication on the details of this method through the Journal of Visualized Experimentation. So if anyone wants any additional details, they're all available there. But just to summarize, in the photos, moving from left to right, in the first step, you are simply brushing scale eggs off of the tree, and then you're putting them on a foam pad and placing them to your test tree. And then a year later, you look on the far right, you peel back the foam, and in the picture is a susceptible tree, and you can see that the insects have established underneath. So once the trees have been confirmed to be resistant to the scale insect, our partners collect SCION for us, and this is shown on the left-hand picture These are our partners in the Pennsylvania Department of Natural Resources and Conservation. So this is very laborious. And by working with partners, it really allows us to take a very cost-effective and efficient approach to getting through this whole breeding program. The picture on the top shows the cut branches that are going to be shipped to us for grafting. We also ask our partners to look for beech trees that are producing seed that we can use as rootstock, because we want to do our grafting as much as possible onto rootstock that's from the same region, because we want to maintain the ability to adapt to that regional environment where the grafted beech are eventually going to be planted. So the rootstock, or the SCION, is shipped to us, and we graft it. And it turns out that American beech is a difficult species to graft. And previously published efforts at grafting beech reported variable success, and it ranged from about 12 to 30%. And it wasn't uncommon for long-term survival to not be very good either. So my technician, Dave Carey, actually went to Hot-Callus grafting, and it was an approach adapted. It was first reported in Filbert. And using this approach, we've been able to increase our success rate to up to 90 to 100%, with our 5-year average being about 67%. The variation is due to genotype. There are some genotypes where they graft very readily, while others are more difficult. But this technique has allowed us to graft a very diverse number of genotypes. So unlike in the nursery industry, where you might be selecting a tree because it is easily grafted, we wanted to be able to include a lot of genotypes in this work. In the picture, you can see that the basic concept behind Hot-Callus grafting is that heat cable that you can see running across the piece of wood in the front. So this whole photo is taken inside of our cold room, where the temperature is kept just above freezing. So we're keeping the rootstock and the SCION dormant, and then the heat cable just warms the graft at the graft union to stimulate growth and to stimulate production of a good union before the tree is breaking dormancy and sap flow starts to occur. So it's been a very successful approach for us. So next I wanted to talk about the next step in the process, which would be breeding, carrying out genetic studies, and then testing. Genetic studies means that you have to be looking at families for patterns of heritability. And you can do this looking at either full sibling families, where you know both the maternal parent and the paternal parent. And in those cases, you have to do controlled cross-pollinations. Or you can use half-sibling families, where you collect seedlings from a specific mother tree, so you only know the mother parent, and the seedlings then probably have multiple different fathers in that situation. Controlled cross-pollinations, we started out doing these directly in the natural forest where the resistant trees were found, and that's depicted in the slide on the left-hand side. And you can see the pollination bags that we put up there to prevent any pollen contaminants from when we actually applied the pollen from our known father onto the flowers of that female tree. We also can do crosses on containerized grafts, and that's shown in the middle photo. So what we discovered is that when we have SCION that was already programmed, the buds are already going to produce flowers. They will still produce healthy, viable flowers that are capable of producing seed even after being grafted. And when we can baby these along and give them great fertilizer and water and whatnot, they actually produce really nice crops of seed with a really successful germination rate. And then the ultimate goal then is on the right-hand side, where you want to be able to have a breeding population or a seed orchard setting that's a little bit easier to work in than your natural forest for producing controlled cross-pollinations for studies. In our first attempt, when we were doing our crosses in the natural forest, we were able to generate 2 full sibling families that are shown at the top in blue. We did a cross between 2 resistant parents and also a cross between a resistant and a susceptible parent. We also collected open-pollinated seedlings from, open-pollinated seed, from a couple of different susceptible trees and from a resistant tree. And the tree that's listed at the bottom here, known as the main tree, it's also a resistant parent tree. The stand that it was located in was actually managed for to enrich for resistance, similar to the cartoon slide that I showed, where the managers went in and removed all the diseased trees. So this tree theoretically would have only resistant fathers around in the stand surrounding it that would be capable of pollinating it. So we collected all these seeds, germinated them, grew up the seedlings, and the next step is to test those seedlings. And we're located in Delaware, Ohio. We do not yet have a natural scale population. It has not hit us yet. So we have been fortunate enough to partner with the Holden Arboretum, and we have our scale screening facility located up in northeastern Ohio, where they do have the scale insect, and a photo of that facility is shown here. So it's been a really nice partnership to have. They've been a very helpful partner. The process for testing the seedlings is similar to that of testing the trees in the field. And again, all the details are in that video and publication I referred to earlier. Basically, you start by collecting eggs off of an infested tree. We actually will sieve the eggs to purify just eggs away from the adult insects and any debris that was collected. In the bottom left picture shows the adult scale insects with, you can see a chain of these oval, sort of oblong, tiny shaped eggs. The females are rounder than they were in the first slide, and that's because they're full of eggs at this point. So we use microscopes to actually count out 150 eggs, and we put them on a smaller piece of foam, and we affix it to the seedling, and we cover it with Tyvek so that as we're watering the seedlings throughout the season, we don't excessively wet the foam and promote fungal growth that can kill the scale insects. And a year later, we come back and remove the pads. We count the number of scale insects on both the pad and the seedling. And in the center picture, you can see it's pretty obvious the trees that are resistant fail to develop a scale population, while the susceptible trees do. In the bottom left picture, I show a highly susceptible tree. So I just want to point out that within the susceptibles, we see an array of susceptibility. Those that are just maybe more tolerant or just slightly susceptible, which support a lower level population, and those where the scale just completely takes off. And in the bottom right, this is showing a picture of an adult that was pulled off with the pad. And you can also see how she's secreting that waxy substance around her and laying her eggs right into that. So our first genetic studies are summarized in this slide. Going from left to right, we are looking first at 1510, which is a susceptible tree. This is the open pollinated population, and 1506, both of which are open pollinated half-sibling families from these susceptible trees. And the blue bar is representing the proportion of resistance. And you can see we're right between about that 1 and 5% that is estimated to occur as sort of your background level of resistance. When we collected seedlings from a resistant tree, the next one over, 1504, we see a slight increase in resistance. But again, this tree, even though it's open pollinated, most of the trees around it are susceptible. So most of the pollen parents would have been other susceptible trees. So it's not surprising that when we actually did a controlled cross between a susceptible and resistant, we're seeing about the same, a little bit better than the open pollinated resistant tree. And then when we look at 1505 by 1504, the two resistant trees that were crossed together, this full sibling family, we're starting to approach 15% or 50% resistance in this population. And the main tree, that stand in Maine, the resistant parent, where all of that is located in the stand where the other trees were removed that were diseased and susceptible, we're even approaching 60% resistance. So when we are crossing two resistant trees, and in the situation with Maine, it's multiple different resistant pollen donors, we're seeing a great increase or a great enrichment in the proportion of resistant individuals in the resulting progeny. So that main open pollinated family gives evidence that in that particular stand, silviculture alone, the removal of the susceptible trees, is leaving behind enough trees that are close enough together to cross-pollinate to just then naturally, in the next generation, create a population that's enriched for resistance. In other cases, the success of that approach is going to depend on, again, the number of resistant individuals that are left behind and how close in proximity they are. So the next step in the process, now that we've done the genetic studies and confirmed that we can enrich resistance through breeding just by pooling together resistant parents, is to start establishing seed orchards. So again, we want to focus on retaining genetic variation. And this slide is just showing the impact of an increasing number of selections on the x-axis in either genetic gain, depicted by the black line, or, I'm sorry, genetic gain is the blue line, or genetic variation, which is in the black line. So to further illustrate this, if we're down at about 5 different clones, we're getting only 30% of the genetic diversity of the population, but that's a 90% gain. So if we're picking the 5 best clones that are highly resistant, we're going to capture 30% of the diversity, or only 30% of the population diversity, but we've really made a big jump in the genetic gain as far as the enrichment of resistance. If we go down here to about 30 clones, and we follow the line up, we're capturing about 90% of the diversity of that population, 95%. So we're typically only going to be losing out on the more rare alleles with 30 different selections, and we're still able to obtain a pretty decent amount of genetic gain, about 70%. So our goal when we're developing our seed orchards is to get between 25 and 30 clones so that we're preserving 90 to 95% of the genetic diversity of the population per region. And I want to stress per region. We're not talking about 25 or 30 clones that are going to be used to populate the entire range of American beech, because we want to make sure within that variation is that adaptive capacity that those trees have developed to adapt and grow in the region that they're in. So we're returning them to their region. So in each region, we want to get 25 or 30 resistant clones. We propagate 8 to 15 copies or ramets of each of those clones. And whenever possible, it's good to have a second replication of your seed orchard at another site, because you never know if something else is going to happen. We actually had a tornado hit one of our plantings. It was an ash planting, but it's always good to have replication, which also allows you to kind of assess how they're doing even in different growing environments. So at this point, this slide is showing all the different seed orchards that we have either in progress, those are the stars that are in yellow, or the different seed orchards that are near completion, and those are the stars that are in black. So if you look in Pennsylvania, we have 2 seed orchards. Those are replicate seed orchards that are almost completed. And then we're starting to work in areas like Vermont, West Virginia. We have a replicated seed orchard in the Lower Peninsula of Michigan that's replicated at Purdue University in Indiana with materials that were collected actually from the Upper Peninsula of Michigan. So we're really starting to get progress and hoping that soon these seed orchards will be producing seed. This slide is showing a picture of a seed orchard at the Holden Arboretum. So the first families that I showed the data on, those seedlings were planted in a research planting at Holden, and we were able to then correlate their initial seedling screen phenotype with their performance after they've been growing in the field for about 10 years. And so we showed that that seedling screen is actually pretty effective in predicting how they perform even when they're growing outside in a plantation, like what's shown in the picture. And over the long term, we'll be able to evaluate the durability of resistance. And on the left-hand side, one of our collaborators from Michigan, Roger Meck, is standing in front of one of our resistance selections. But Holden has also now committed to, they've gone in and removed the susceptible trees of all of those families, and we're working with them to increase the diversity by adding new selections to this planting. So once these trees start to produce seed, and we're also using those containerized seed orchards to do a bunch of crosses and produce seedlings to provide material for outplanting. And this is we're just at the beginning phases of being able to have enough material that's enriched for resistance to start doing outplantings. And at this point, we actually only have one, and that's shown in this picture. Our partners at Michigan Department of Natural Resources planted 138 seedlings, about half of which are resistant to beech bark disease, back in 2011 at a site that was heavily impacted. You can see some of the downed beech trees in the background there by beech bark disease. So at this point, we know we have about 80% survival of those seedlings, and then we're going to be working with them to track their progress and to try and identify ways, learn more about what it's going to take to get these seedlings back out in the forest. So I've mentioned my partners several times, and I just put together this flow chart to give a summary of sort of the network, the tree improvement cooperative that we've put together. And if there's anyone listening that would be interested in participating with their forest that has a beech bark disease problem, please feel free to contact us. Again, our state and national forest partners are doing the surveying in the field, they're testing in the field, collecting the SCION, and then we at the Northern Research Station are actually doing the grafting, the breeding, and the genetic tests. The Oconto River Seed Orchard has also joined us in helping us with the grafting efforts. We carry out the testing with our partner Holden Arboretum. Once we have the grafts available, they go out to our partners to establish the regional seed orchards. We'll help them with that process and with designing the seed orchard, but then after that, they take care of all of the maintenance and upkeep of those seed orchards. And eventually, once the seed orchards are producing seed, they will then distribute seed to their state and national forest nurseries for germination to produce these seedlings enriched for resistance that can be used in outplanting. And then just to summarize, again, wanted to point out this idea of using breeding to improve host resistance is not a new concept, and it's something that can be applied to other invasive species issues. And just to review some of the pros, it may be the only feasible solution. And think of chestnut blight, for example. And through a breeding program, you can actually help with conservation of a species, preventing it from becoming extinct, as well as the other species that may depend on that species. I know in the case of chestnut, there was different moth species that were documented to go extinct after the decline of chestnut. There's a proven record of success of this approach. Public support is usually pretty strong because it's that green solution. It's sustainable, little or no side effects. On the con side, resistance it can sometimes be very rare. With beech, that 1 to 5% is actually more on the high side. So it can be pretty labor intensive to identify that resistance. Obviously, this is a longer term approach when you're talking about breeding, so it's going to require a long-term investment. And sometimes your level of resistance is not as strong as the phenotypes that we see in beech bark disease, so it requires a more complex breeding strategy to increase that resistance. So sometimes there's an uncertain outcome. But overall, I would say that it is an approach where the pros are outweighing the cons. And that is where I was going to end. Again, I'm just acknowledging the fact that we have a huge network of collaborators that have helped us with all of this work. Acknowledge our funding sources, that none of this would have been possible without. And then in the photo, the picture of all the members of my lab group. So I would be ready to take any questions now if there are any.
Well, thank you, Jennifer. We do have a question. Dr. Sadoff said, given that the scales are immobile, is there a chance they could eventually adapt to the host plant resistance you select for in your orchards? Do you think that having regional seed orchards at with 30 plus resistant lines could reduce the chances of this happening?
Yeah, that is definitely one of the reasons why we want to make sure we're retaining genetic diversity. This is not a single gene resistant trait. We have done some work with mapping and identifying different regions of the genome that are associated with the resistance trait. And so we've identified 2 major gene loci and at least 3 minor gene loci. And that's just in a single mapping family. So given the variation in our phenotypes of susceptibility to resistance, and we even see variation in the resistant phenotype, in that some trees, we see no adults establishing. Other resistant trees, the adults can establish, but they fail to reproduce and then eventually die off. So keeping that variation helps prevent that insect from overcoming the resistance.
Okay, that seems to be the only question we have at this point. But I am kind of interested when you're talking about starting the tree improvement programs. Is the main kind of reason that they kind of died out or aren't doing anymore? Is it because of funding, or is this something that somehow we could, people that are doing research on this could somehow find ways to have more cooperators, or what do you see as some of the ways we could see more of these tree improvement programs starting?
Yeah, I think the tree improvement programs were sort of driven by the economy, and it was a commodity. So timber is a commodity, and of course the Forest Service and other federal agencies were supporting tree improvement programs, but a lot of the big cooperatives were groups of industries working with universities and other researchers. So one of the problems was that the investment wasn't always necessarily giving you more earning potential on your product. So that led to the decline. And then just the general sort of decline in federal funding, I think, over the years has always also contributed to that decline. Now, the problem with invasive species, especially when you're talking about a tree like beech or butternut or some of those where you lack a specific market and you don't have that commodity piece to it, then yeah, it's going to depend a lot on government funding or organizations like the American Chestnut Foundation that have private donors. So I would say that people that are interested in supporting it, one of the best things to do would be contact Forest Service officials, let them know you think it's an important approach, let your congressman know to support breeding.
Okay, that's good. I'm just kind of wondering about that, because of the ash tree. Everyone is talking about ash trees going extinct, but we're still, like you say, we're finding some of these ash trees that are a little more resistant than others, and thinking about that too, because like we all kind of know, this probably is not going to be the last invasive wood boring problem or disease that's going to happen. So this has been very interesting for me, especially
right.
Seeing the how you build this program and everything. But like you say, it just the time and the money, it kind of looks like those are kind of a couple of limiting factors, as many things are with these kind of issues. So
well, a couple of things. But from the time we started working with beech till the time that we started actually getting those seed orchards established was about 10 years. So when you think about how long emerald ash borer has been here now, If we had been taking a genetic approach the whole time, we might be farther along. But I can tell you, we are actually working with ash, and we are breeding and applying the same process to it. So it's not necessarily always the slowest process. It just isn't a good fit with your typical 3 to 5 year grant cycles. So it can be kind of tricky to piece together that continuous funding to support that kind of work.
That's good to know. That's interesting. I wanted to let you people know that there is the survey for the EAB University. I've put that up. That's probably not going to be a live link for you, but I just wanted, if you wanted to copy it down, that's fine. Again, I will be sending out this link in an email that will be, so the link will be live, that you can get into the survey and let us know what you think. Is there any other questions while we're here? We've got a couple more minutes, but if there's not, I don't want to take up too much time for everyone. But I wanted to thank you, Jennifer. This has been very interesting, as all these EAB University webinars are for me. It just kind of shows me a whole nother way of how folks are trying to deal with some of these problems that are going on our woodlands and forests and, urban forestry, that kind of thing. So I see something is here. One more thing here. Oh, I see someone, Cliff has said, thank you so much for your presentation.
Yeah, thanks for having me.
Yeah, and with that, I guess, unless you can always get a hold of Jennifer, I'll send out her email as well if you have questions afterwards. But I wanted to thank everyone for participating. I'm sure we're going to see more folks looking at the recorded webinar. And our next webinar is going to be February. Let me grab my calendar here real quick. Is going to be February 11th at 11 a.m. All our webinars are on the emeraldashborer.info website as far as what's going to be presented and when. So I encourage you to look at those too. Thanks again, and I will be closing the meeting.
Thanks, Robin.